Hello, thank you for joining. As part of the ARA and PigRegen funded project, we have prepared short presentations with the main outputs from experimental trials. Research was done by the monogastric research group, and most of the trials conducted were performed at the AFB pig farm in Hillsborough. For the current presentation, we have Dr. Anna Lavery, that she will be presenting the outputs of two trials that she conducted within this the ARA and PigRegen funded project. Hello, Anna, and I pass you the presenting rights.
Thanks, Ramon. So can everyone see the screen okay? Yeah?
Yes.
Perfect, okay. So this presentation will cover two studies that looked at sow nutrition to improve early piglet performance. So this work was carried out as part of a larger project which aimed to improve the number of pigs produced per sow per year. And from the diagram, you can see a number of factors contribute to sow productivity. And much work has been done with genetics to look at litters per sow per year, and as well as nutrition and management techniques to minimise the non-productive sow days. So this work was really going to focus on more of the left-hand side of this diagram and look to improve the number of piglets born alive, and also to minimise pre-weaning piglet mortality. So to do this, we look towards nutritional strategies, and in particular towards the sow. So previous research focusing on maternal nutrition has shown some really promising results with improvements in fetal development, the number of pigs born alive, and piglet growth as well. So the overall aim of this work was to complete two studies. The first of all was to look at nutritional strategies during gestation to improve piglet vitality at birth and growth to weaning. And then our second study focused on nutritional strategies during lactation to support milk production and piglet growth until weaning. So our first study looked at salmon oil inclusion and vitamin D3 level during gestation. So why salmon oil? Well, additional fats are often added to pig diets to increase the energy density, but they actually also have great nutritional benefits. So salmon oil is a rich source of omega-3 fatty acids, but currently there are no recommended inclusion rates for omega-3 oils in sow gestation diets. But previous research with salmon oil, which is high in DHA, which is an omega-3 fatty acid, has seen improved piglet vitality and vigour at birth, improved piglet growth until weaning, and reduced pre-weaning mortality rate. So when we're looking at vitamin D3, due to the indoor housing systems, supplementary vitamin D is essential for pigs to maintain calcium metabolism, bone health, and immune function. And the D3 form, called calciferol, is commonly used in pig diets, and it doesn't depend on this light dependent stage, which is shown on the right here. So while current recommended inclusion rates from the NRC are for 800 international units per kg for sow diets during gestation and lactation, levels of up to 2,000 international units per kg are commonly used in commercial diets. And the reason for this is that vitamin D is a relatively cheap vitamin to include, but 2,000 international units is really the upper limit that can be supplemented over long periods of time. So previous research in this area has documented some benefits of including up to 2,000 international units per kg, and that included reduced stillbirths and improved birth and weaning weights. This isn't surprising, as during gestation and lactation, the requirement for vitamin D3 increases with the demand for calcium, which is needed for fetal growth and milk production. So our study objective was to see if including salmon oil and the 2,000 international units per kg of vitamin D could improve piglet vitality and viability at birth and reduce piglet pre-weaning mortality. So this first study was carried out between May 2016 and April 2017 and involved a total of 120 multi-pair sows and just over 1,800 piglets. The dietary treatments started on day 30 of gestation and until farrowing, and we wanted it to start on day 30 basically to target early skeletal development of the piglets. And prior to the trial starting, all sows received trial diet 1, which contained soy oil and 2,000 international units per kg of vitamin D. So all the diets were formulated to contain the same energy, protein and lysine content and were cereal soya based. And our oil comparison then was soy oil versus our salmon oil, and then our 2 levels of vitamin D. So all the sows on this trial were allowed to farrow naturally, and we didn't induce any of the sows. And this was due to the fact that it's thought that fish oils and salmon oil included in that can extend gestation, and that's maybe why some studies didn't see improvements in piglet vitality. So we took all the normal measurements at birth, such as total born, born alive, and born dead. But the additional measures taken depended on whether the farrowings were attended or not. So we observed 80 sows farrowing, and the measures taken at birth included birth order, vitality score, birth interval, blood lactate and time to first suckle. And then at 24 hours, we did some more measures such as ponderel index and BMI, rectal temperature and IUGR score. So a bit more detail about these measurements. The vitality score we used involved watching each piglet for 15 seconds after birth and scoring it between 0 to 3, 0 being no movement or breathing, and 3 being good movement, attempts to stand, and the piglet is breathing. We also measured blood lactate, as this is meant to give a measure of hypoxia. And we did this by getting drops of blood from the umbilical cord immediately after birth and testing it. And we used a blood lactate monitor, which is similar to a diabetes blood glucose monitor. So at 24 hours, we measured each piglet's crown to rump length and abdominal circumference. So we could look at ponderel index and BMI, which have been shown to have links to survivability. So piglets that are disproportionately long and thin are often born later in farrowing and have decreased survivability. We also give each piglet an intrauterine growth retardation score, which is the IUGR, which has 3 different levels and includes traits such as the dolphin-like shaped head, bulging eyes, and wrinkles at the side of the mouth. And the IUGR score has also been linked to colostrum intake and survivability. So the sows that farrowings were not attended were used for milk samples and pig dissections. All the piglets in the litter were weighed, and a piglet of mean birth weight was selected for dissection. So we collected blood samples, brain and liver samples, and we used these for fatty acid and vitamin D analysis. And we also took hind leg samples to look at bone strength and bone mineral content testing. We also collected colostrum samples and milk samples again at day 14, and they were also analysed for fatty acid and vitamin D content. So looking at the results then, so with regards to all the sow measures, the only differences that we detected was a difference in lactation intake. So sows offered the soy oil diet in gestation at 12 kilos more in lactation than those sows fed salmon oil. And there was no significant effect of treatment on any other sow or litter variables measured. So the mean total born in this study was 14.9 and born alive was 14.4. We standardised all the litters to 14 piglets within 24 hours of farrowing. So the average litter size after that was 13.7 and our mean number weaned then was 11.4. So when looking at piglet vitality measures, so we had just over 1,100 piglets from the 80 litters that were used for vitality measures. and we didn't find any interactive effects between the oil type or the vitamin D level on any of the piglet vitality measures. What we did find was that there was a greater probability of piglets receiving a mild IUGR score, a greater Ponderella index and BMI score if born to sows offered a diet containing soya oil compared to that of salmon oil. When we were looking at the farrowing, then we had a mean birth interval of 13 minutes and time to first suckle was 22 minutes. When we looked at birth order, piglets with a low birth order, so were born earlier in the farrowing process, they actually had a greater probability of receiving a higher vitality score. So that was to be expected. But what we did see, which is shown here in the graph, is that time to first suckle was actually shorter for piglets born later in the farrowing process. So just over 1,800 piglets were used to evaluate growth performance to weaning. So what we did find here was the piglets born to sows offered a diet containing soya oil and a low level of vitamin D3 gained almost 29 grams a day more than sows offered a diet containing soy oil and a high level of vitamin D. Now here we have the straight effects of oil and vitamin D. And we did find the piglets born to sows offered a diet containing a high level of vitamin D3 were actually heavier on day 1. But when looking at gain later in the lactation period and overall weaning weight, piglets that were born to sows offered a low level of vitamin D3 during gestation gained more in the last 2 weeks of lactation and as such were heavier at weaning. When we look at the fatty acids then, as expected, the fatty acid concentrations in blood, milk and tissues were influenced by the dietary oil type fed. So feeding a diet containing salmon oil increased the total N3 fatty acids and decreased the total omega-6 fatty acids in sow blood plasma, colostrum and milk compared to a diet containing soy oil. What we have here in the table is piglet plasma results. So total polyunsaturated fatty acids and omega-6 fatty acids were higher in the plasma of piglets at birth when they were born to sows offered a diet containing soy oil. If we then look at piglets born to sows offered diets containing salmon oil, total monounsaturated fatty acids and N3 fatty acids were greater from piglets born to sows offered diets containing the salmon oil. So as a consequence of this, there was a greater proportion of N3 fatty acids and lower omega-6 fatty acids in piglet liver, and a total increase in polyunsaturated fatty acids and omega-3 fatty acids in piglet brain tissue as a result of feeding salmon oil. So when we're looking at vitamin D3 levels, so here we're looking at the 25-hydroxy D3, which is the circulating form of vitamin D3 in blood. So on the left-hand side of these graphs, we have the vitamin D results from sow plasma. So feeding a high level of dietary vitamin D3 increased 25-hydroxy D3 in sow plasma compared to feeding a lower level, which was to be expected. If we're looking then at the right-hand graph here, this looks at the vitamin D3 and the 25-hydroxy levels in colostrum and milk. So sows offered a diet containing a high level of vitamin D3 during gestation had an average of 2.1 and 1.2 nanograms of vitamin D3 and 25-hydroxy D3 respectively in colostrum and milk samples. And when we looked at piglet liver samples, the high level of vitamin D3 during gestation increased both vitamin D and the 25-hydroxy-D3 form in piglet livers at birth, compared to those born to sows who were offered a low level of vitamin D3 in gestation. We did analyse piglet blood plasma samples for 25-hydroxy-D3 levels, but these were actually found to be below the detection limits of 3 nanograms per mil. So the take-home messages from this study: the type of dietary oil and vitamin D3 level did influence fatty acid and vitamin D status of both the sows and piglets, but this didn't translate to an improvement in piglet survivability or growth to weaning. We did see that piglets born to sows offered a low level of vitamin D3 diet during gestation, were about 200 grams heavier at weaning, and this may require further investigation, as we did use a 2000 international level of vitamin D in the lactation diet, so whether this had some influence on that needs investigated. But we also finished these treatments in the study at farrowing, so it would be interesting to continue these treatments through the lactation period also. So the second study that we completed focused on lactation nutrition strategies, and we looked at salmon oil inclusion in the diets, but also a phased energy regime. So with the salmon oil, previous research has shown that oil inclusion in diets during late gestation and lactation not only influences the fatty acid composition of milk, but also increases the fat and the energy content of milk and improve piglet gain then to weaning. So again, there's currently no recommended inclusion rates for N3 oils in sow lactation diets, but previous research using fish oils has seen an increase in the DHA concentration in both colostrum and mature milk, which improve piglet growth, and also been associated with improved piglet growth post-weaning. When we're looking at the feeding regime, it's estimated 70% of the sow's lactation energy requirement is destined to support lactation, and so it's important to maximise sow feed intake. If feed intake has already peaked, then it's increasing the energy content of the lactation diet may better support the demands of the litter and minimise the loss of sow body reserves. So when sow feed intake commonly plateaus in late lactation, it was thought maybe using a phased feeding approach in late lactation might improve energy intake, and we did this through an increase in dietary energy content by increasing the oil inclusion in the diet. So the aim of this study was to investigate the effect of replacing soy oil with salmon oil in the lactation diet, and also increasing the energy level from day 15 of lactation. So this study was carried out between June 2017 and January 2018 and involved 100 multi-pair sows and just over 1,100 piglets. Dietary treatments were started on day 105 of gestation, and that was due to previous research which reported colostrum and liquid droplets detectable in mammary tissue of swine from day 105. So what we looked at was then a comparison of soy oil versus salmon oil and the flat versus phased feeding regime. So our flat feeding regime was a 14.5 megajoule per kg digestible energy diet for the whole 28-day lactation period. And then our phased feeding regime was the 14.5 megajoules per kg digestible energy diet until day 14 of lactation, and then it changed to a 15.5 megajoule per kg digestible energy diet until weaning at day 28 of lactation. And these diets were offered in meal form. So just some of the measurements then that we took. So we looked at sow weight on entry and exit of the farrowing house, sows, the back fat depth and body condition score were measured regularly throughout the study. The vitality measures were somewhat scaled back in this trial, but we looked at litter suckling duration and frequency in the first 24 hours post farrowing, and we recorded individual piglet weights regularly. We also collected colostrum samples from sows and collected milk samples again on day 14 and 21 of lactation. So looking at the results, we didn't see any significant treatment effects with regard to sow back fat or body condition score or weight. But as expected, sow back fat depth, body condition score and weight decreased as lactation progressed. And so we had an average decrease of 3.2 millimeters in back fat depth and 16 kilos in weight from farrowing to weaning. We did see that sows offered the diet containing salmon oil at 3.5 kilos more during week 3 of lactation, but overall we didn't see a difference in feed intake. But as expected, feeding a higher energy level from day 15 to weaning increased sow energy intake during this period, but also in the overall lactation period. When we were looking at milk yield in the study, it was calculated as piglet gain times 4.2, and that was a calculation from previous research. So we find an effect of oil on sow milk yield. Sows offered the diet containing salmon oil produced on average 1.2 litres a day more from day 7 to 14 and 1.7 litres a day more from day 21 to 28 than sows offered lactation diets containing soy oil. We didn't actually see any effect of the energy regime. But what we did see, independent of the diet treatment, was a day effect. So milk yield did decrease on day 5, but increased again on day 7, which you can see in the graph. And milk yield peaked at day 14 and then plateaued to day 28 of lactation. So when looking at litter performance, we had a mean total born of 15.7 in the study and a born alive of 14.1. Again, all litters were standardised to 14 piglets within 24 hours, and so the mean litter size after fostering was 13.8. The average number of piglets then weaned was 12.2, and pre-weaning mortality rate had an average of 11.2. So there was no interaction between oil type and energy regime for any of the litter performance measures we collected. So litters from sows offered salmon oil during lactation had an increased litter gain in week 2 and week 4 of lactation compared to those from sows offered diets containing soy oil. And this isn't surprising when we've seen these effects also with milk yield. Sows offered a diet actually containing salmon oil had a tendency for a reduced pre-weaning mortality rate compared to sows offered diets containing soy oil, and this was a value of 9.9 versus 13.4%. When we looked at litter suckling duration and frequency in the first 24 hours post-farrowing, it didn't differ between the dietary treatments, and we didn't see any effect of energy regime on any of the litter performance measures to weaning. So when we look at colostrum and milk fatty acids, the dietary oil type significantly influenced the fatty acid profile of sow colostrum and milk samples. So as a result, the proportion of total saturated, monounsaturated fatty acids and omega-3 fatty acids were increased in milk samples from sows offered diets containing salmon oil compared to those fed diets containing soy oil. We also seen a significant interaction between oil type and energy regime in late lactation on the fatty acid profile of milk that was collected on day 21 of lactation. So offering sows the salmon oil and a phased energy regime increased the proportion of omega-3 fatty acids and decreased the ratio of omega-6 to omega-3 fatty acids. Whereas when we look at soy oil and the phased energy regime, there was an increase in the proportion of total polyunsaturated fatty acids and more omega-6 fatty acids. Irrespective of the sow dietary treatment, during lactation, there was a significant effect of day on the fatty acid profile of colostrum and milk samples. So the proportion of total polyunsaturated fatty acids, omega-3s and omega-6 fatty acids were greatest in colostrum and decreased by approximately 50% in milk at day 14. But when we looked at day 21, these all increased again, and that was due to the fact that we had our phased energy regime at that point. So what were our take-home messages then? In this study, we didn't see a synergistic effect of the type of oil included in the diet or the energy regime on sow or piglet productivity. And there was no improvement in sow condition or piglet growth when we were using the salmon oil, but we did see a tendency for decreased piglet mortality and an improved milk yield. So therefore, salmon oil in sow lactation diets should be investigated further, as they could increase piglet survivability and growth through an increased milk yield. The lack of treatment effect in this study may be a result of the fact that there was a high sow lactation feed intake. So this also demonstrates how important it is to improve sow lactation feed intake to support the modern sow. So thank you for your attention. I would like to acknowledge the funders of this project, DAFM and the University of Leeds, all the OptiPig project team that were involved, as well as the AFB Pig unit staff for all their help and guidance through all the trial work. So if you have any questions on this work, please get in touch via the email, and the details are on the slide. Thank you.
Thank you, Anna, for your presentation. And just a quick question on the first trial that you present. You mentioned that the vitality score was higher for piglets that were born earlier, but the time to first suckle was actually longer for them compared to the ones born later. Do you know, or do you have any idea why this would be the case?
Yeah, well, this was something actually we observed because we were sitting there doing all the observations. And we think later in farrowing, with the other piglets already suckling, the piglets born later in the farrowing process can hear their litter mates suckling. They can smell the milk, the colostrum, and so they actually were able to locate the teats far quicker than those first piglets born, who actually spent quite a bit of time trying to find the teats and start to suckle. So we think that was part of the issue.
And then a second question on the second trial, that you found no effects on back fat condition and back fat body condition of the sows. Did you look at the interval from weaning to insemination or subsequent fertility?
No, we didn't, unfortunately. In hindsight, I think that would have been really interesting to look at, and maybe is something that we can go back on, because we would have that kind of data recorded as normal practice on the farm, but it isn't something we investigated.
Okay, thank you. And thank you all again for joining this presentation.